Great, thank you. So, hello everybody, my name is John Cromaldi. I am the lead PI on the new Odor to Action NeuroNex network. And as such, I have this great privilege of working with a team of 16 really incredible scientists from around the world. And as a team, we're really excited to get spinning up on this new collaboration. And I just wanted to take a quick minute to say how incredibly impressed I've been by not just the quality of the science talks and posters over the past few days, which I kind of expected, but in particular, really especially impressed by the thoughtful discussions on the various dimensions of diversity. So, you know, a shout out to the NeuroNex3 organizers and NSF and the other funding agencies for not just for making this meeting happen, but for providing, to use their words, sort of a new dimension to a science meeting that I know, especially in some of the small breakout rooms, has been really stimulating, and I've learned a lot. So that's been great. So in this session, we have 9 pre-recorded talks. The first 3 talks are overviews of the 3 IRGs in the Odor to Action network, and then there's 6 more science talks from a range of other NeuroNex projects. So as always, a reminder to please post questions in the chat throughout the talk so we can have a lively discussion afterwards. And if possible, please be explicit about who your question is directed to in the chat, so that'll help down the road. And as we move towards queuing up these video talks, if I could just take my session chair hat off for a quick second and just put my Odor to Action network hat on to give a very brief overview of the network as a way of introducing the first 3 talks. So the Odor to Action network seeks to understand how animals use information from odor stimuli to guide natural behaviors. And we have 3 IRGs, and those will be the first 3 talks. IRG 1 is interested in the generation of neural representation of that odor stimuli. Then IRG 2 is interested in how neural circuits then lead to subsequent behavior. And IRG 3 is interested in how those active behavioral responses actually change the way in which the animal perceives the odor. So why don't we go ahead and queue up the talks, and then we will have question and answers after all the talks are done.
Hi, I'm Betty Hong, and my lab works on odor processing in the fruit fly brain at Caltech. We're fortunate to be part of the newly launched Odor to Action NeuroNex network, and I'm the lead PI for IRG1. We're an international network comprised of 16 labs funded by 3 agencies across 3 countries. Our network asks, how do animals use chemical information in the environment to guide natural behaviors important for survival? We emphasize a few key approaches. First, we focus on natural stimuli, natural and chemical composition and in spatial temporal structure, and on natural behaviors. Second, we believe in a comparative approach across organisms. Olfaction is an example of a more general problem that occurs in many complex neural systems. How are high-dimensional, discrete, and combinatorial variables that are not easily ordered along obvious axes represented in the brain? To understand the relationships of how odors map across these 3 spaces shown here, IRG1 will test the hypothesis that they're organized around the statistics of odorants and natural odor sources important to the animal. We'll work in 3 animal models: the fruit fly, the honeybee, and the house mouse. And IRG1 is comprised of 6 investigators: myself, theorist Vlad Itzkov, Hiro Matsunami, who works on the molecular genetics of mouse olfaction, computational neuroscientist Tanya Sharpie, Brian Smith, a honeybee behavioral neuroscientist, and Matt Wakowiak, who works on olfactory coding in mouse. IRG1 is motivated by the idea that due to the shared biochemical processes that produce volatiles in natural sources, the abundance and co-occurrence of specific molecules has a hierarchical organization that should relate to the behavioral relevance of the odor source. In other words, the odor space of the natural world is structured, and we think the brain takes advantage of this structure. Hierarchical relationships are hard to capture with linear methods, and theorists Tanya Sharpie and Vlad Itzkov work with non-Euclidean methods that better capture these relationships using alternative geometries. They'll apply these methods to embed odorants in natural chemical space, neural representational space, and behavioral space, and we expect these geometric models will reveal new organizational axes important for olfaction. On to our specific aims. We'll use the chemical profile of lots of natural odor sources to construct the natural chemical spaces as I described. In flies and bees, we'll test whether the principal axes of natural odor space map to important behavioral variables. For instance, Brian Smith's group will extract axes in natural floral chemical space that map to the nectar and pollination status of flower blooms. He can construct mixtures that then vary systematically along these dimensions and evaluate their predicted ability to impact bee foraging behavior. On the neural coding side, the first step is to understand the representation of odor space by the repertoire of odorant receptors and how they form a spatial map in the brain. In mouse, Hiro Matsunami is using molecular profiling and spatial transcriptomics to expand the spatial mapping of olfactory channels in the brain, collaborating with Matt Łakowiak on functional validation. In mouse and fly, Matt's lab and my lab will be systematically expanding neural maps for large numbers of natural odors and natural mixtures at successive stages of olfactory processing. Ultimately, our goal is to use different geometric models of neural space at each processing step to test how each relates to the odor space organized around natural statistics, or alternatively, whether neural spaces might be better described by geometrical relationships organized around other odor properties like physical chemical features or behavioral value. And of course, IRG1 functions within the larger network, and we'll be closely working with the other two IRGs in our network to achieve an end-to-end understanding of how natural odor structure impacts olfactory behaviors. We're very much looking forward to interacting with the other NeuroNex networks. If there are interesting synergies, please get in touch with us, and thanks for listening.
Hi, my name is Nathan Urban. I'm a professor of biological sciences at Lehigh University, and I'm part of the Odour to Action NeuroNex grant, and I'm the lead on IRG2 entitled Sensory-Motor Integration. The focus of IRG2 is to address the question of how is odor sensation translated into natural motor behavior. As part of this, we're looking at the decomposition of a variety of different odor-guided behaviors, and then the mapping of elements of those behaviors onto particular circuits in the brain. So for example, we're looking at behaviors such as odor-guided trail following, such as shown on the left, and looking at how it is that particular turns as part of that trail following are controlled. Or, for example, looking at circuits in the fly brain that are responsible for detection of wind direction, which is an important component in that particular odor-guided behavior. For these experiments, we're looking at a couple of different animal models. We're looking at adult and larval fruit flies for those experiments, and we're also looking at mouse. The team here consists of myself, Tomoko Oyama from McGill University, Kathy Nagel from NYU, Lisa Stowers from the Scripps Research Institute in San Diego, and Bard Ermintrout from the University of Pittsburgh. There are a number of different components of this project that come together and integrate with the other IRGs. In particular, we're proposing to look at many aspects of the statistics of odor-guided naturalistic behaviors using a variety of different approaches, machine learning techniques that have been developed over the last few years to analyze pose, position, motion, and to categorize those behaviors. We're doing this in the context of odor-guided navigation and also of a set of odor-related social behaviors. Taking the data from those different approaches, we are constructing a variety of different agent-based models of these naturalistic behaviors and using these agent-based models as inspiration for understanding and analysis of which particular circuits in the brain we think will underlie these behaviors, what kinds of properties those circuits must have, and that will allow them to mediate those behaviors, to combine behaviors in a variety of different ways. And in addition, we're looking at specific sensory circuits on the one side that extract the features of the stimuli that are needed to perform these behaviors, and also looking at specific motor circuits that underlie those actions. By combining these analyses of circuits at the sensory and motor level with the circuit level modeling, we hope to be able to provide an explanation for the behaviors that we see. Oops. Sorry. With that, I'd be happy to take any questions.
Hi, my name is Brian Smith. I'm the lead PI for IRG3 in Odour to Action with a focus on active sensing. Our research activities will focus on studies of the dynamics of odor dispersal in plumes, using theoretical models for evaluating strategies for sensing odors in different plume structures, and how animals' movements, and in particular their sensor movements, can enhance the detection and localization of those odors. We'll use two different animals in our research, the honeybee which uses antennal movements, for example, as well as body movements to localize odors, and then mice, which have who sniff as well as use body movements as they orient in odor plumes. These animals have different types of sensors, possibly employing different strategies that we intend to evaluate in the process of our research. We'll also incorporate the use of robots in our models in later years as we begin to understand some of the strategies for orienting in plumes, and we try to test those strategies by implementation in robots. There are 6 of us in IRG3. 3 of them are behavioral neuroscientists working with honeybees and mice. There's myself, Andreas Schaefer at the Francis Crick Institute in the UK, as well as Justus Verhagen at Yale University. 2 theoreticians will be integral to the activities of our group. One of them is Jonathan Victor at Weill Cornell Medical College, as well as Michael Schmucker at the University of Hertfordshire in the UK. Finally, John Cromaldi is an engineer who studies odor, plume, and flume dynamics using various kinds of structures for creating these plumes and flumes in his laboratory. We'll begin with the use of wind tunnels in Cromaldi's lab to study the plume dynamics of natural odor scenes. And you've heard already a lot about the types of odors that we're going to be studying. And we will study initially how those odor scenes are created under different kinds of conditions of turbulence in wind tunnel situations in the Cromaldi laboratory. That information will be integrated, for example, fed into computational models that investigate different theoretic strategies for detection and coding of odor sources in plumes. And these model predictions will be evaluated using behavior, electrophysiological recordings, and optical imaging in honeybees and mice. And the outcomes will be iteratively fed back into the modeling. Finally, AIM-3 will integrate virtual reality environments into the whole experimental protocols we're using, and then evaluate how animals use the information to localize plumes, and then those experimental results will be fed into the development of robots. The 3 IRGs are coordinated around the use of natural odors that are appropriate for the natural histories of the different animal species. And importantly, across all of the IRGs, the integrated work will use insects and mammals that share common structural elements to their olfactory solutions, to the solutions to olfactory problems. And the team-based coordinated investigation will lead to potentially the discovery of some fundamental principles of olfaction. Thank you very much. We would enjoy your questions and comments.
Well, good afternoon. Thanks a lot for the opportunity to present our work and program here. My name is Andreas Schaefer from the Francis Crick Institute and University College London, and I'm part of the Odor to Action NeuroNex network. My lab contributes, among others, to IRG3, active sensing, as you just heard from Brian, studying the dynamics of odor plumes and in particular, how active sampling shapes odor representation and behavior. My contribution is based on prior work from my lab where we have shown using whole cell patch recordings in mice during learning that active sampling profoundly impacts on odor representation and the discriminability of stimuli. So mice adapt their active sampling strategies from early to late in the learning process to improve odor representation. In this Odor to Action NeuroNex project, we will investigate how sniffing affects odor representation in actively exploring navigating animals using Justus van Verhagen's olfactory virtual reality system. Natural odor plumes, however, are highly spatial-temporally complex structures with rich temporal structures in the way odor concentrations change at frequencies of tens to hundreds of hertz. Here within the network, John Cromaldi, with many other parts of our network, will measure odor plumes and how sniffing shapes these odor plumes. In prior work funded by the Wellcome Trust, my lab has begun studying such high-frequency odor plumes, and for that we have developed an odor delivery system that is capable of reliably presenting well-controlled odor fluctuations at frequencies as high as 50 Hertz or even above. Using an automated behavioral training system, we have shown that mice can detect astonishingly high frequencies in odor fluctuations. So now in the NeuroNex project, together with Justice van Berhagen, we will integrate this presentation device with the olfactory virtual reality to mimic the very plumes John and others are actually measuring. In order to more broadly then read out neural activity, we will contribute to the NeuroNex network a new large-scale electrophysiological recording technique that we have recently developed with Brain Initiative support. Here we are combining CMOS chips with microwire bundles, thereby lifting sort of the scalability of the CMOS architecture to the third dimension for neural recordings deep in the mammalian brain from dispersed neuronal populations. This will then allow us to record from large populations of neurons in different olfactory centers, for example, olfactory bulb or anterior olfactory nucleus or further down olfactory cortex, and then describe how active sampling in a navigation task impacts odor representation. And with that, I want to thank my colleagues in the network and all of you for your attention. Thank you very much.
Hi everyone, my name is Maribel Patiño. I'm a graduate student in the lab of Ed Callaway, and today I will be talking about the development of a new rabies-based circuit tracing strategy named START, which stands for Single Transcriptome Assisted Rabies Tracing. So the goal of START is to allow us to identify transcriptomic cell types making local monosynaptic connections onto specific classes of Cre-expressing cortical neurons. This method combines Cre-dependent monosynaptic rabies tracing technology using a G-deleted rabies virus that expresses a nuclear fluorophore with fluorescence-activated nuclei sorting and single nuclei RNA sequencing to allow input neurons to be assigned to specific transcriptomic types. So before moving forward with this project, we first needed to assess whether rabies infection was compatible with single cell sequencing and transcriptomic cell type identification. We know that rabies virus infection is known to influence gene expression, but we didn't know the extent to which it does this and whether it would prohibit the classification of cell types. Thus, to use single cell sequencing to accurately characterize rabies infected cells, it was necessary to conduct transcriptomic cluster analysis of large numbers of rabies-infected cortical neurons and identify the correspondence between these clusters and establish transcriptomic clusters from uninfected neurons. So here I show that with the use of computational strategies for integrated analysis of heterogeneous single nuclei RNA sequencing datasets, it is possible to align and integrate rabies-infected nuclei with uninfected cortical nuclei. And using integrative analysis, rabies-infected and uninfected cells grouped together by established cell types compared to a control experiment without integration where nuclei grouped primarily by their dataset origin and infection status. Thus, despite rabies-induced changes in gene expression, it is still possible to assign rabies-infected neurons to established transcriptomic cell types previously identified. We can then use this combined data set of infected and uninfected neurons as a reference atlas, which can transfer cell types labels to query input cells from rabies circuit tracing studies to allow for transcriptomic characterization of rabies-infected input neurons. And I'd be happy to take any questions. And thanks to everyone in my lab and all the help from the Salk Institute and the Allen Institute.
Hello, my name is Monica Gonzalez, and I'm a graduate student in Evan Miller's lab at UC Berkeley. Our lab develops fluorescent chemical tools for the purpose of studying membrane potential. Today, I will share with you how we have used a phosphine oxide rhodamine voltage reporter, abbreviated PO-ROVER for voltage imaging. Like other voltage reporters, synthesized in our lab, PO-ROVER increases its cellular fluorescence with increases in cellular membrane potential. However, unlike previous voltage reporters, PO-ROVER absorbs at 700 nanometers and emits at 727 nanometers, making it the reddest voltage reporter to date. Pullover stains the membranes of cells as shown in the image on the right, and using patch clamp electrophysiology, we determined that pullover has a 17% fractional change in fluorescence per 100 millivolt change in cellular membrane potential. After bath application of pullover to dissociated rat hippocampal neurons, we're able to record spontaneous activity. Pullover provides us with excellent spatial resolution, allowing us to look at the activity of the neurons in our field of view. If we zoom in on an area of activity, we can determine through the timing of action potentials the direction of communication. Through these recordings, we can visualize action potentials with great signal-to-noise ratios and spatial temporal resolution. Moving on from cultured cells, we began to characterize pullover in mice retina. This RD1 retina lacks photoreceptors, making it mostly unresponsive to light. However, the remaining layers of cells still exhibit spontaneous electrical activity. We imaged voltage activity alongside GCamp, which was expressed in the tissue. To confirm that pullover faithfully reported voltage changes, we compared the recordings to those of a multi-electrode array. The electrodes are the black circles seen in the images. The fluorescence traces were obtained from the ROI drawn in blue around the electrode. From the traces, we see that the retina is active and that the signals from the MEA and pullover precede the calcium signals from GCamp as expected. If we zoom in on a region of the trace and invert the MEA signal, we see that it overlaps perfectly with the MEA recordings, confirming that pullover reports changes in voltage. From these recordings, we can also analyze traces from the areas between the two electrodes. The images on the left show rectangular regions of interest that correspond to the traces on the right. If we focus on a portion of the trace, we see that between the ROIs, activity is diminished in ROI 6 and 7 compared to ROIs 1 through 5. If we chemically block receptors, we can inhibit synaptic transmission. And this results in large rates of activity that are homogeneous across the retina, possibly done using gap junctions. This experiment shows that we can track changes in activity in response to pharmacological manipulation with greater resolution that is not possible with an MEA. With that, I would like to thank my lab and Professor Evan Miller, as well as our collaborators in the Kramer Lab. Thanks to the organizers for allowing me the opportunity to share our NSF research, as well as NSF for funding. Thank you.
So neural circuits are wired specifically to process information and encode behaviors. And to study how circuits form and change over time or in disease, it is crucial to easily visualize synapses in live animals in a high throughput manner. So our hub is using the invertebrate C. elegans as a model for answering key questions in developmental neuroscience, with ample precedence of mechanisms of wiring being conserved in organisms across phyla. So the good thing is that the entire connectome of C. elegans is well mapped and established, as is shown in the slide here, but it is still constrained by a small sample size and conditions. So since the project started, our hub has developed and disseminated several tools in the C. elegans community. These tools include synaptic reporters, such as those shown here, made using various technologies such as split fluorescent proteins and iBlink for chemical synapses, and then CRISPR and Cas9 to endogenously tag electrical synapses or gap junctions more recently. Our goal is to label many edges in the C. elegans wiring diagram to help ourselves and others answer key neuroscience questions such as how the connectome varies among individuals, under different genetic backgrounds, across developmental stages, and under environmental stressors, but in a high throughput manner. And through our efforts and those of other labs, I can say that we've collected a wealth of synaptic reporters, and we continue to generate more, but there exists no central database to standardize image collection, annotation, and sharing biological data and imaging metadata. So this is where Hang Lu's lab comes in. Their goal is to standardize image collection and analysis to facilitate comparisons across different users, different experiments, labeling techniques, and research labs. And currently they're working on such a central database or repository, you could say, which is built on the open microscopy Omero system. We also wanted to go a step further and standardize the process of synapse quantification so that anyone using these reporters or their own reporters could infer biological information from huge data sets in an unbiased manner without spending many, many hours and having multiple users scan synapses to remove biases. And on this end, the Hobart Lab has worked together with members of Lu Lab to develop a tool called Cycle, which stands for Punctate Synapse Counter. This would automate image analysis. Psycho uses a unit-based deep learning framework, and it is trained using existing data to process large image data sets that are collected by diverse users in diverse labs and using diverse microscopes. And this software is able to cluster images in an unbiased manner into bins based on synaptic features or phenotypes. So we envision the pipeline would allow a naive user to let the computer do the job, which is very time-saving. But it also allows a more advanced user to specify additional parameters and extract more information than would be possible with the naked eye. The pipeline also resolves technical problems that have been long-standing in the field. For example, background fluorescence, complicated neural morphologies, dense synaptic regions, all of which confound synapse quantification. So currently we're in a phase where we're reaching out to more and more labs across the globe to test our pipeline and give us feedback. And we're also putting together a manuscript to share the first version of it with the Worm community. With this, I'd like to thank members of the Hobart Lab for generating strains, members of the Lou Lab for building the pipeline, and NSF for funding. Thank you. I'll take questions now.
Hi, my name is Adam Kepecs at Washington University right now, and I'm going to tell you about a formal language for behavioral task description and execution. And the work was done by Michael Wolf, an engineer in my lab, and Maryam Majeed remains at Cold Spring Harbor, a postdoc. And we have a collaboration to extend some of these ideas with the NWB, the Neural Data Without Borders project, with Oliver Rubel and Stephen Edwards at Columbia, who is an expert in real-time languages. So what's the challenge that we addressed? We believe that the behavior is really a bottleneck for neuroscience, whereas progress in neural measurements have been really science fiction-like. Progress in behavioral measurement and quantification have been limited. And specifically, there's no language to define behavioral tasks. So that means that when you design a behavioral task, it is difficult to share, and reproduction is impossible unless we use the identical hardware-software setup. And moreover, task design is hampered. So our goals were to design a language that is sufficiently expressive to describe all behavioral tasks, describe real-time event contingencies, and it's easy to formalize and translate into machine code so you can actually execute it. So it's not just a drawing, but really the actual code that runs. And there's additional benefits to this, which is to expose the behavioral task logic that ensures the clarity and precision in task design. So you can think about a behavioral task sort of at a physical and an algorithmic level. At the physical level, as an experimenter will deliver stimuli, and the subject will take actions, and these can be described formally and measured. The algorithmic level is what we addressed, which is that an experimenter sets up program contingencies really to use it to infer the brain algorithms. And so how do we describe these program contingencies? So we have this language now, we call it BETL, or Behavioral Task Description Language, that comes as a diagram and as an XML code. And this is just an example to see for a perceptual confidence reporting task that my lab uses. On the left, you can see a human understandable version of how you see a particular trial unfold with some code snippets on the top. And on the bottom, you can see a piece from the XML code that formally describes the same thing. And now with this in hand, we have a pipeline for how we can take the XML code and turn it into execute it on a piece of hardware. We're still working on a design environment, but once you have the XML description, which we have for many tasks already, we have a BETL parser that translates it into a control environment. Right now we have this in MATLAB, and in the future we plan it for Python as well. And then translate it into hardware. My lab uses BPOD, which we developed, and it's an open source project, so you can use it. Over 70 labs use it, including the International Brain Labs collaboration. But you can design it code generation for other target hardware, so that's not limited. And so now we can take an actual task description and turn it into a running protocol on a computer. And we're working with a number of case studies to show the generality of this, as you can see above. And right now, we have this ready. So if you're interested, please let us know, and we can work with you to formalize your behavior. And you can see the poster by Michael Wolf to describe more details about this project. Thank you.
Greetings. So my name is Christopher Moore, and I'll be telling you about our bioluminescence hub. In the hub, we're generating new optimal tools for both control and imaging of dynamic active cellular networks, obviously of neurons. These tools have a variety of advantages that are not covered by other methods. Now, the way we do this is we have a closely knit team of collaborators going all the way from molecular innovation to in vivo testing. Because of the short time, I'm going to focus on existing tools that are already working and used in a variety of papers, and that we've now made a really transformative advance in by working across our team. So the main way that we try to control cells by using bioluminescence is the approach called BLOG, where we use local bioluminescent light created by enzymes within a cell to drive optogenetic elements. Now, typically the way we do this, although not exclusively, is by creating luminopsins or LMOs, where we actually tether the enzyme that produces the light to the optogenetic element. Now, what are the advantages of using a luminopsin? Well, there are a variety. One of the most important is it provides tremendous flexibility experimentally. It works both as a chemogenic agent. You inject a small molecule and that creates bioluminescence in any cell expressing it, or the same molecule, the same luminopsin, can be used only for its optogenetic properties and providing very precise spatial and temporal regulation. Another kind of advantage of this approach is that every time there's a drug interaction, a chemogenic interaction, bioluminescent output light is generated. So you actually can literally track the efficacy and timing of your control system in real time. Now, a recent advance made in Nathan Shearer's lab has been to make much brighter bioluminescent elements. As you might imagine, increased photon output is increased efficacy. This is one such example, ECAL9H. When we incorporated that into the luminopsin construct in what we're referring to as LMO7, the evolution of LMO3, in vitro, we get approximately 8-fold greater induced subthreshold currents over LMO3, which again is already being used effectively in vivo. When we look in vivo at LMO7, it generates about an order of magnitude brighter light. Here we see a comparison of LMO3 and LMO7 from mouse sensory neocortex, and produces very robust multi-unit activity, about a 3-fold increase in practical terms over LMO3. These methods work not only by direct application of the drug, for example, in this case to the neocortex, but also by IV injection. So our priorities in sum are to advance existing tools to make them brighter and stronger, as I just showed you. LMO7 will be out soon, and you can contact us if you'd like to use it. We're also making a variety of new kinds of calcium sensors. One reason we're doing that is that BL light is great for one-photon imaging. Another reason is that then we can create activity-dependent regulation of optogenetics through calcium fluctuations driving light production. One technology that we've just recently really gotten to work nicely is what we call interluminescence. And it's an example of activity-dependent regulation with light, where we've created essentially an optical synapse by putting the optogenetics on the receiving side and activity-dependent bioluminescence on the presynaptic side. So replacing normal chemical transmission with photons across the synapse.
Hey, well, welcome back, everybody. Fun to see all those great talks. So we have some time here for Q&A, and there's been a few questions that have been posted in the chat, and I would encourage you to keep putting them in there. I think it's nice if I can just call on people to actually ask their questions themselves, and a reminder to unmute yourself when I call on you, and also show yourself if you can. Joshua, do you want to go first? You had a question for Adam.
Sure. So Adam, your beepod and beetle and bee other stuff looked really cool. I wonder if it can be applied to honeybees, for example, like with their odor stuff from the odor to action group.
Yeah, basically, it depends on, there's another question, what is a task? A task is a set of contingencies. Animals take actions, you have measurements, and you do something else. You deliver a new stimulus, you react, you change things. Anything that can be described such a way can be described in XML, but obviously you need a piece of hardware to run it. So our BPOD project, it's open source, and so far it works out of the box by compiling it onto that hardware, but this is not a limitation. You can, if you have another piece of hardware, you can compile it onto that as well.
Okay, great, thanks. Let's see. Cosmer, you had a question, I think, for Christopher Moore, correct?
No, it's for Adam as well. It's just a follow up. So would it be possible to use this setup to compare behaviors across related species? For instance, frequently same experiments are run on different primates, primate species, so humans and other primate species. So would it be, could it be used for that?
That's exactly the kind of thing that we're hoping this is going to be used for, is any of those tasks can be described the same way. And in fact, sometimes it's a problem. You describe a task as a working memory task, but the details are so different. And that's why it's not enough just to describe those details. You need to kind of execute it, because if you don't execute it, you never really know if your description fits the actual ongoing timing of the task, which is often critical. So yes, we basically, there's no limitations in this way. You can compile it into any hardware for any species. And certainly the primate tasks that have been used are exactly the type of tasks that this is for.
Okay, going to the next question here. Let's see. Yusaku, you had a question for Monica.
Yes. So for the voltage sensor, how fast does the signal rise and decay? I'm guessing it's like millisecond time scale. And second one is, have you tried 2-photon excitation? If so, how does it work?
Hi, thanks for your question. So since the fluorophore is a small molecule-based probe, it's pretty fast. So it just depends on the rate of fluorescence, which is in the nanosecond range. In terms of the 2-photon imaging, we can do that. The only limitation is how fast the microscope can scan and take the images. So really, we're only limited by basically the imaging technique.
Julio, do you want to ask your question?
Yes, my question was more related to Adam. When you talk about the language and the sensory motor tasks, I mean, it's clear. But what we call in mental representations, those are things that are floating around in the mind, and it doesn't actually produce the behavioral output many times. Can your language be expanded to represent this and maybe create a little bit of this realm that we see, especially in primates, mostly macaques or humans?
I mean, can you give me an example of how would that work in your case?
For example, you can have different alternatives in a working memory task in which you have 3 or 4 objects, but only one of them is going to produce some kind of output. But still, you have to hold the representation of the different objects in mind. But you have to mentalize. You have to actually, without necessarily acting.
So this belongs to sort of the realm of the brain algorithm, right? So if I have this physical and algorithmic level and the experimenter and the animal. I mean, as neuroscientists, I think that's what we were all trying to do is infer the brain algorithm, the mental representation. But I don't think anything can be sort of directly captured at. The critical thing about inferring the mental algorithm is that the physical algorithm that's running is described with in the same framework. Often what we end up doing is running a task, and then when you apply a model, you don't apply it to the actual behavioral contingencies. You apply it to your interpretation of it.
I see.
And that gap can be often really, really important if you're, for instance, trying to infer a reinforcement learning algorithm. The actual timings could be critical. So that's what we're trying to fill, and it's all used sort of to figure out how that other box works, how the mental representations and the brain algorithms work once you have this the contingencies formalized.
Roger, you had a question for Brian Smith.
Unmute. Okay, if I understand correctly, the Odour to action network is going to resolve an understanding of how there's different animals, different size, different time scales, how they solve this similar problem. You mentioned they have similar strategies, but they have different sensors. So is it the idea that you'll find out how they do things similarly and how they do things differently, so it's going to be a comparative analysis?
Yes, I think that was one of the objectives, and then we can take some of these strategies and actually implement them in robots to evaluate them. Your question was different lengths and different time scales. If by that you mean going from, and this is sort of my question to you, going from, say, bacteria, orienting in a chemical gradient, to C. elegans, orienting in chemical gradients, to elephants and blue whales. I mean, do you mean that kind of length and time scale, literally?
No, I see you're looking at insects and small mammals, right? So at least they're significantly different. Those two animals are significantly different. You mentioned that they have similar behaviors, but different. You also said that they had different sensors. So I'm wondering if you have a guess at the differences that they're going to be using in the circuitry based on that information, and will there actually be significant difference in behavior when you look at it closely?
No, I don't have a guess at that. How they use that information differently in the more central circuitry, say, if that's your question. That's something we want to try to find out by me working with Andreas and Justus, for example. They work on mice, I work on honeybees. And I'm interested in how honeybees can modulate their antennal movements in a way that may enhance their ability to pick up odorants in the same kinds of plumes that the mice are using changes in body position and sniffing. And whether that translates into the same kind of information, say, in the antennal lobe, which is the analog to the olfactory bulb, I think that's something we really want to find out. But of course, when you see these differences, then the big question will be, when we try to take them and you work with Michael Schmucker to implement them in robots, can we use that as a test bed for the hypothesis that you're talking about? But I mean, I said bacteria to blue whales. I mean, I wasn't really thinking of that scale difference, but it's an interesting question, I think. Where an animal with their head movement can be operating, you know, the amount of their environment they sample is very different. And, you know, you have bacteria tumbling as well. I think it's an interesting question that we can start to think about.
I know somewhat from working with Mark Willis and Moz. I know something about how they'll do casting and
that's right,
spiral kind of movements in normal odor tracking, and so they do a lot of body movements where when they're flying they can do this, where when you're walking, a small animal walking, you're limited on how those kind of movements, I suppose.
Well, you're limited. You don't have 3 dimensions when you're walking, obviously, but a cockroach orienting to sex pheromone in a plume will show the same kind of casting and surging behavior that a moth will show, or a mosquito or a honeybee will show in 3 dimensions. Thanks. I mean, it's an interesting sampling problem when an animal no longer is getting or picking up odor filaments, and we were talking about this with Jonathan the other day, the modeler in our group. When an animal surges upwind because it just detected a plume, well, how long does it have to go before it says, I need to now cast, right? And you'll even see in moths and honeybees, they'll surge upwind, and instead of immediately moving back and forth, they'll hover for a few seconds, right? And that's a sampling problem. And that's an interesting sampling problem. Is something that we hope to be able to address. And by the way, say hi to Mark from me.
I will do so, yes.
I hope that addressed your question without too much hand waving.
Yes, it does. And I look forward to the future results.
Nathan, did you have anything you wanted to add to that?
I mean, I'll just jump in that, I mean, certainly this is an issue that we've thought about in a couple different contexts. So if you think about the sort of spatial and temporal scale of fluctuations in odor concentration for a fruit fly walking versus a mouse sort of moving through an environment, both based on sensors, an array of sensors, and also based on body movements. They're doing a very different thing in terms of what it is that they can, how it is that they can sample that environment, and then how it is that they make use of that sort of differential sampling, or how that differential sampling influences their behavior is something that we're definitely interested in exploring. We have talked about sort of other species where the separation between sensors is even is larger than any of the ones that we've just mentioned, as another potentially interesting set of examples to look at. So these are all questions that are near top of mind for us, and I think it certainly relates to some of the work that your group is working on, your network is working on with respect to some of these issues of movement as it relates to common features of movements as they relate to different spatial and temporal scales.
Jonathan, do you want to jump in on that?
Scale,
go ahead, Tom, then Jonathan.
Oh, I'm sorry. At an even finer scale, the Dictyostelium, amoeboid form, single-celled organism, has an ability to sense, a gradient that is so shallow between one end of the animal and another, a concentration gradient of single molecules of odor, of substances, their body is so short relative to the concentration gradient that, it's hard to see mathematically how they can even achieve what they obviously do achieve. So there are some really interesting, molecular mechanisms, signaling pathway mechanisms inside the Dictyostelium that are used to make these computationally through a reaction pathway make this kind of very sophisticated discrimination.
Yeah. Actually, I keep thinking I'm done talking, so I mute myself, and then a question like this comes up with Dictyostelium, and we're talking about scales.
Yes.
We had a discussion the other day when a moth or a bee is orienting in a wind plume, it's picking up filaments from the turbulent nature of the plume. So it's sort of like you pick up a filament, you surge up wind, you don't get one, you cast until you get another one. And we had a discussion, well, are they picking up concentration? And I would love to ask Mark this question, Mark Willis, because I bet he would say filaments are more important than concentration gradients in that situation. Whereas at the scale of a Dictyostelium or bacterium, concentration might be the more important thing to try to pick up.
And actually, they're picking up single particles. So the whole concept of concentration is a red herring.
Right.
So it's really about how many collisions with an odorant molecule is occurring per unit of time, perhaps.
And in some ways, the filament example is like that sort of single molecule, because it may be collisions, if you will, with single filaments on the one scale, and collisions with single molecules on another. And so this idea of sort of of chemical being delivered almost as in a Poisson process or inhomogeneous Poisson process kind of way is one. Bard, if he's on, has built some of his agent-based models on these kinds of approaches rather than on a continuous concentration gradient kind of model.
Yeah, it's a stochastic sampling problem.
All right. Well, sorry to jump in here and interrupt, but that was a great conversation. We've gone a few minutes over time. I know we were given a little bit of latitude since we started late, but I think so that we can get out of here on time today, we'll turn it back over to the organizers. So thanks, everybody. I really enjoyed that.
Junior colleagues who presented and spoke up for the first time in this meeting. I was excited to hear from you, and I know it takes some guts to speak up in a large crowd, so thank you for doing that. I also want to thank, before I give the final closing remarks to Flo, just thank Flo and Sri and Reed and everyone at NSF, Jim and Namo for supporting this effort. It's been in planning for many months, pre-pandemic era. And I've been honestly very worried about it, which is pretty unusual for me because I usually don't worry, but I had no idea what was going to happen. And I'm feeling really relieved that it seemed to have gone very well, and I learned a lot. So I'm really grateful to all the participants that made it a success. I'm particularly grateful to Linnea, who really spearheaded so much of the intellectual thought in planning all of this, and to Sarah and Kelly for Sarah organizing and doing everything. We had many, many late night meetings over the last several weeks and months. And so for that, I'd also like to really thank my wife, who we have 2 small children, and while they were waking up in the middle of the night, she was getting them, and I was working on this, including the last 2 days where I've been engaged fully in this, and she's been engaged fully with them, which is an incredibly hard and important task. So I'm grateful to her. And I don't know, Linnea, do you have any final remarks you'd like to say before Flow closes?
No, I'll just echo all of that. Tremendous, tremendous effort by Joshua and Sarah. Really impressive to pull this whole thing together. And thank you all. It was so thrilling to have so much participation in all aspects of the meeting. You guys really make the meeting. I think this went beautifully. So a hand for all of you too. And thanks to the session chairs and the speakers.
Panelists.
And on behalf of the NSF, can only echo what Linnea and Joshua already said. Really hats off and many, many thanks to Joshua, Linnea, Sarah, and Kelly, to all the session chairs, and importantly to all of you for having participated in making this a real interactive meeting. We didn't know how interactive it would be. And along with Joshua, yes, I was panicked a little bit too, and Joshua had to put up with that. So it seemed like you really took advantage of the different ways in which to interact, which is fantastic. I'd like to also echo some of the words that Joanne Torno mentioned yesterday, namely to really try to take the information that you learned here, the discussions you had here, to not leave them just in the space of this meeting. This is a start, if you wish, or for some of you, a continuation, and continue it outside of the walls of this meeting. Take it back to your institutions, take it back to your hubs, teams, networks, groups, and really engage. And you heard the voices from the trainees. They want to engage, so really make this an interactive activity across all levels, all institutions, the many dimensions we discussed. There was, integration not just in terms of the science, but also integration in terms of the sociodynamics of the scientific enterprise. And then I also would like to echo words that I heard in one of the discussion rooms, and that is, team science is really, there are a lot of opportunities. Yes, I heard it in multiple rooms, many opportunities. There are challenges, which you discussed, and there will be new challenges around as you develop it further. But think collectively, you're the network of networks that the NSF is trying to develop here in the context of this program. So think constructively about it, talk to one another, talk to us, how we can realize this, what may sound utopic at times, but if you only work, if you work for it, then you can actually make it happen. If you don't if you don't strive for it, then you're not going to make it happen. And team science has a lot of potential. So share with us our concerns and our excitement about it. And we look forward to seeing you in about a year from now. So it's for those who are new to NeuroNex, we always try to link the annual meeting to the SFN meeting, and whether it's in person or virtual, we'll see you for sure next year. And thank you again to the organizers and to Sarah and Kelly.
Thanks, everybody. Enjoy your day or your night, depending on where you are.
Thank you all.